Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.
Well, it's an honor to be asked to speak at this series that's been going on for some time. When I got the call, a message was left on my telephone answering service to come and speak at Saturday Morning Physics, and I thought surely they must have the wrong person because I don't really do physics, but they said no, in fact this is what we would like you to do. So as was pointed out, I'll be talking to you about, this is the second in the series on this trilogy on the cheery topic of weapons of mass destruction, and my assignment is to talk about chemical weapons, and I have a subtopic here called chemical agents as triggers of neurological disease, which I'll be getting into toward the end. Here's an outline of what I'll be talking about. First of all, just to reacquaint you with what we mean by weapons of mass destruction. It's not an easy term to define, and people have different definitions of this term. Go into chemical weapons, review that in general, and then move into the specific topic of nerve agents and their target enzyme, acetylcholinesterase, or ACHE, and then move to neuropathic agents, which is the topic of my own area of research, and the target for these called neuropathy target esterase, abbreviated NTE, and how that's related to neurological diseases, both chemically induced and possibly idiopathic neurological diseases. And then just a brief conclusion and some indications of future work. Well, what are weapons of mass destruction, or WMD? I think WMD a couple of years ago was cited by a couple of institutions as the most hackneyed phrase of the year, or the word of the year, overused, misunderstood, and so on. But there, at least in the United States, there's some agreement among government agencies that we're talking about agents or munitions that can kill or incapacitate large numbers of people and do this in an indiscriminate manner. Now, these could include conventional weapons, the usual munitions that blow things up or burn things up, incendiaries or explosives. But they've come to mean unconventional weapons. And we have this trilogy that marks this series in the Saturday Morning Physics talks of NBC, the nuclear, biological, and chemical weapons. But we also are now including radiological weapons as distinct from the nuclear explosive weapons or nuclear weapons. And here are the three symbols of these, and this is me right here, the skull and crossbones. Well, you've probably seen, or many of you in this audience, I think, are in the right age group to know Cary Grant in the movie North by Northwest, a landmark film of Hitchcock. And I put this slide in to ask a question: what is unconventional warfare? Now, in this case, is Cary Grant, whether he's Kaplan or Thornhill in the movie, being chased by a crop duster, this biplane that's loaded up with probably DDT. The film was made in 1959. They were probably still using DDT in those days. And you may recall later in the sequence, he does get dusted with a white powder that probably was something like DDT. Well, is this chemical warfare? Is he the object of a chemical warfare attack? Well, in some sense, he is. And this notion of what we mean by unconventional weapons goes back to the military scientist Clausewitz, writing around the 19-teens in a famous book called On War. And in that book, Clausewitz talks about the objectives of different sides in a dispute or in a battle, their strategies, what are their targets, and what kinds of weapons they might employ. If you're sort of the underdog, if you're not as sophisticated militarily as your opponent, You may try to gain the advantage by winning hearts and minds of people or by scaring them to death as a means of achieving your objective, which may not be a military objective, but a political objective, an ideological objective. And the main point here is that the object of your attack could be one person, it could be a group of people. It doesn't have to be mass destruction, but the idea is to create fear. You can see that Kerry is a bit worried here. He's running for his life. And this kind of unconventional attack has made a great statement. You know, we could have gotten you in other ways, but we're getting you in this quite unexpected, rather bizarre method. In a more serious vein, here we have Alexander Litvinenko, who, as you know, was in the news recently, his death due to being poisoned by, we now know, the isotope of polonium, polonium-210, which is really a radiological weapon. So it combines radiation with chemistry as a delivery system of radiation inside cells, where it can be very effective at killing at very low doses. It's estimated that the LD50, or the amount needed to kill a human being, to kill 50% of the human beings in a population of humans, is on the order of 50 nanograms of polonium-210. And the lethal dose for one person is, the sure lethal dose is probably around 0.89 micrograms of polonium-210. So extremely potent weapon. And one of the things that whoever was responsible for carrying out this murder, was to demonstrate their potency of using a somewhat elaborate scheme. You know, we could assassinate people in conventional ways, with a pistol, for example, but doing it in such a way demonstrates our potency. It arouses mystery. There's a certain mystique associated with this kind of weapon. It combines terror with the actual objective of going after what you perceive to be your enemy. It displaces the notion of having control over your environment. I mean, we still are arguing about how Mr. Litvinenko got this polonium-210. And essentially what it does is to create dread. I talk in my lectures in toxicology about dread space, the 4 quadrants of dread space. And this installation of dread has to do with, it's a function of our understanding of a hazard and our perceived sense of control over it. And our dread will be exaggerated beyond the actual risk if we feel that we have little or no control ourselves over the exposure, over the managing the risk. We may or may not understand a lot about the risk, but the sense of loss of control scares us. And so again, the bottom line is that we're trying to instill terror and intimidation by using unconventional weapons. This scene is from World War I, where weapons of mass destruction, chemical weapons, were first used on a large scale. And here we have a cloud of chlorine gas that the British troops are running through with their gas masks. You can see that they're not really terribly well protected against things like nerve agents, as we'll see in a moment. You know, it looks like they might be barehanded. They do have gas masks and goggles, but it wouldn't offer a whole lot of protection against some kinds of toxic agents. The definition of a chemical weapon, you could say that a chemical weapon is, well, anything. If you make a plutonium bomb or uranium bomb, that's a chemical. So nuclear weapons could be chemical weapons. Conventional explosives could be chemical weapons. But they've come to mean chemicals that use their toxic effects rather than their incendiary or explosive properties to produce the damage. They also depend on a chemical interaction between the toxic agent and the biological molecules within us, and so they must gain access to biological sites of action. This is very important, that one way you can avoid harm from, for example, this cloud of toxic gas is if you're back here in the woods somewhere, you're not going to be exposed. So exposure is critical. The stuff has to gain access to your body in some way, penetrate the skin, get into the lungs, be in the GI tract, and get absorbed into the blood and taken to tissues where it can exert some damage. Now here we have a sophisticated chemical sensor, this hapless rabbit in a cage, where the man is very well protected against the possible leakage of the nerve gas sarin from these canisters. This is a U.S. facility from around about 1970, when we were still working on chemical weapons as weapons, and not strictly for defensive purposes. And they used animals as essentially sniffers of whether or not we had a leak, because these canisters are filled with the nerve gas sarin, and if there is a leak, the rabbit will start to show symptoms and possibly die, and then the man hopes that there's not a breach in his protective suit. The types of chemical weapons that we have, this is just a small assortment of them, and they're categorized in different ways by different people. But there are blister agents that are vesicating agents that cause blisters on the skin, actually by a very complex mechanism. It's not a matter of just something that's corrosive, that sort of burns on contact, but it actually has to do with a long string of biochemistry, an attack on the DNA, and that's not the subject today, so we won't dwell on that. So-called blood agents that don't really attack the blood, but really attack the cells. Blood is the delivery for these agents, and it's maybe highlighted by the agents hydrogen cyanide, very toxic gas, that essentially blocks the ability of the cell to utilize oxygen. And so you die fairly quickly from hydrogen cyanide. There are so-called lung agents like phosgene that irritate the lungs and can cause death from the lungs filling with fluid. And later there can be, if you survive that, there can be severe scarring in the lung tissue. And then one of the subjects today are the nerve agents, and I'll be using as an example the nerve agent sarin, which is given the designation GB. You may have heard these designations VX, GB. GB is the one that's used for the nerve agent sarin. Now, I think we're all familiar with this scene from the news back in 1995 when a terrorist branch of a religious cult in Japan decided to use fairly low potency sarin in the Tokyo subway system. It ended up poisoning thousands of people and killing a few. And this is a scene from that episode in 1995. The signs and symptoms of poisoning by nerve agents like sarin, and by the way, this was, as I said, it was reckoned to be a low potency sarin agent that wasn't distributed terribly well, or the carnage would have been much, much worse. But here we have some of the things that can happen if you absorb a toxic or a lethal dose of sarin. There'll be salivation, sweating, lacrimation, urination, defecation, which together make up the acronym SLUD, S-L-U-D. It's not a pretty way to die, if there is a pretty way to die. Meiosis, pinpoint pupils. I'm very fond of giving the derivations of words in my classes, and one day when I was talking about this, I told the class that this is from the Greek goddess of pinpoints, Myos, and everyone dutifully wrote that down. It may be true, I don't know. But, then there's rhinorrhea, profuse watery runny nose, bronchorrhea, excessive bronchial secretions, bradycardia, the opposite of tachycardia, slow heart rate. And by the way, you can have a fast heart rate during part of the poisoning episode, and some people will respond differently with maybe tachycardia instead of bradycardia. So this isn't a universal thing. If it's a high dose, you can go into coma, respiratory arrest, and then death. But even if you have all of these signs and symptoms, before death, you can be pulled back from the brink by certain antidotes that we have available now, and certain treatments that we have available for this kind of poisoning. Well, how can an agent like sarin do all of these things? How can it produce this laundry list from SLUD to death? It's kind of like reading the package insert for your bottle of pills from the pharmaceutical company. The package insert will say anything can happen from rash to death. And it's sort of like this, how can all of this happen? Well, it happens because of action, very specific action, on one enzyme in our nervous systems called acetylcholinesterase, ACHE. Sarin inhibits this enzyme, as we'll see later. It works at cholinergic synapses cholinergic meaning synapses, connections between nerve cells and their target organs that use the neurotransmitter acetylcholine as the chemical messenger. Works both in the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which is all the nerves emanating from the central nervous system. Subdivisions of the peripheral nervous system are the autonomic and somatic nervous system, carrying out automatic functions and carrying out voluntary functions. And we'll say a little bit more about that as I go along. The morbidity and mortality that you see from sarin toxicity is really due to excess of your own neurotransmitter. You actually end up killing yourself from an excess of your own transmitter, acetylcholine, because it's not being broken down by acetylcholinesterase. And before we get into this mechanism, there's a little review that's called for, a little review of the nervous system. And here we have a motor neuron. And I mentioned that this is not drawn to scale, and I'll show you this in a second, and why this becomes important. But all nerve cells, you can identify certain anatomical parts. There's the cell soma, the cell body, that's the region immediately around the cell nucleus. And here we have all these branches called dendrites. There's one long process that's specialized for carrying an electrical action potential, for carrying essentially an electrical wave from the cell body out to the tip end of the axon, where we have this terminal bouton, the synapse. There's not a direct electrical connection between the ends of nerve cells and the next nerve cell or a target organ such as a gland or a muscle. The electrical signal is transduced from an electrical signal into a chemical signal where it interacts with receptors on the postsynaptic membrane of this other cell over here, triggering an action potential in that cell if the signal was big enough, and setting up this cascade of nervous system or neuromuscular or neuroglandular action. We also have a myelin sheath, which is a bit like electrical tape wrapped around this axon in order to provide some electrical shielding to prevent crosstalk. It's an insulator to prevent crosstalk between axons as they're carrying out their business. Now I've got a little high-tech demo here, and this may be difficult holding the microphone and holding a basketball. I wasn't known for my coordinated skills in sports. But if we try to get the scale of this neuron, if we think of the cell body, if we shore off the dendrites, the cell body is this basketball. Okay? This is the scale. And our axon is this bit of hose. Imagine this is a garden hose. And we attach this to the basketball. And over here, at the end, we have something that looks like maybe a sprinkler. It would mimic this, the terminal branching of these axons and the synapses coming out of the end of this garden hose. Well, on this scale, the garden hose coming out of this basketball cell body would have to be about 1.5 miles long. So if we're sitting here in Ann Arbor, the synapse for this garden hose axon would be over in Ypsilanti somewhere. So that gives you an idea of the scale. We're talking about a cell body that in our bodies is something about on the order of maybe 100 micrometers in diameter. We're talking about an axon that might be maybe 0.1 or so micrometers in diameter, but it could be 1 meter in length or 1 million micrometers in length. So when you do the math, you find out that if you roll up this length of garden hose, that you're going to have a volume that is 100 times the volume of the cell body. So whenever you see these textbook drawings of nerve cells, they're not properly to scale. So keep the basketball and 1.5 miles of garden hose in mind. This becomes important when we talk about insults to the axon later and what it means to the life of the neuron. Well, here's a highly schematic view of the synapse, this place where the signal is transduced from electricity into chemistry and then back into electricity as the signal crosses the synapse. This gap is on the nanoscale. It might be on the order of maybe 20 nanometers from here over to here, from the presynaptic membrane to the postsynaptic membrane. Now this represents a cholinergic synapse. It might be something like the synapse between a motor neuron in your spinal cord and a muscle in your leg. But all synapses have essentially the same kind of machinery, and they have to carry out the same kind of business, whether they use norepinephrine or acetylcholine or something else as a transmitter. There has to be synthesis of the neurotransmitter, in this case acetylcholine, made from acetyl coenzyme A and choline, and an enzyme to catalyze that reaction. They have to be stored and packaged in synaptic vesicles, and that's these little circles here. Each vesicle holds what's called a quanta. This has nothing to do with quantum physics, but a quanta of neurotransmitter molecules, which is around about 10,000 molecules. And those of you who remember Avogadro's number from elementary chemistry know that that's really a very small number of molecules to be dealing with. There is a release mechanism as part of this transduction of getting the acetylcholine out of the vesicles and into the synaptic cleft. So the vesicle essentially merges with the presynaptic membrane, expels its contents into this small cleft, and then diffusion takes over, and these molecules simply diffuse across to the other side, where they interact with receptors that, in the case of a nicotinic receptor on your muscle cell, is a sodium ion channel. The acetylcholine causes that ion channel to open. Sodium is higher in concentration outside the cell than inside the cell. The sodium will then flood into the postsynaptic cell following its concentration gradient. And if there's enough sodium coming in, it will trigger depolarization of the postsynaptic cell. And if this is a muscle over here, it will cause that muscle to contract. If it's a gland, that gland will secrete something. If it's another nerve cell in the central nervous system, that nerve cell may fire and propagate this chain of activity. There has to be a way of, we've now activated these receptors. We've now turned a switch on. There has to be a mechanism for turning the switch off. So we have to inactivate the acetylcholine. And that's where acetylcholinesterase comes in. These little red boxes over here represent acetylcholinesterase molecules on the postsynaptic membrane. They are like biochemical hoovers that are vacuuming up excess acetylcholine and regulating the concentration. See, all of this is a concentration-driven mechanism. If the concentration is high enough, we'll activate these receptors. If it's kept low, we won't activate enough receptors to cause any action over here. These acetylcholinesterase molecules can be inhibited by organophosphorus compounds such as serin, and that's illustrated here. Serin is blocking the acetylcholinesterase. If we don't have acetylcholinesterase chewing up these acetylcholine molecules at a sufficient rate, we'll build up a high concentration of acetylcholine in the synapse. All of these receptors will be triggered, and we'll have firing of the postsynaptic cell, contraction of the muscle, secretion of glands, firing of other nerve cells. Everything is going off at once, and we're essentially overstimulated by all of this cholinergic activity, and if the dose is high enough, we die. Acetylcholine is quite pervasive in the nervous system. This is a sort of engineer's view of the nervous system laid out in blocks for you. Here we have the central nervous system, the autonomic nervous system, peripheral nervous system, parasympathetic autonomic and sympathetic autonomic nervous system. And then the somatic, where we carry out voluntary action. I want to click the mouse, so I reach over here. That's the somatic nervous system. Let's start with the somatic. We have a motor cell in our spinal cord that's connected directly through a chemical synapse to a muscle. And the synapse there is called the neuromuscular junction, the NMJ. So we have acetylcholine as a neurotransmitter. In all of these, what are called preganglionic sites of connections emanating from the sympathetic and the parasympathetic nervous systems in the autonomic nervous system, we have acetylcholine being used as a transmitter. And on target cells in the parasympathetic nervous system, we also have acetylcholine used as a neurotransmitter. Over here, from this ganglion, we have norepinephrine used as a neurotransmitter, but that's driven at this ganglion by acetylcholine. So in all of these loci throughout the nervous system, we have acetylcholine being used as a neurotransmitter. The viscera are controlled by the parasympathetic and sympathetic nervous systems. Sympathetic is part of the fight or flight syndrome, where you stand and fight or you run away. The parasympathetic is sometimes called the rest and digest system. And if you have all of these going off at once, there's just chaos. And this explains some of the symptoms, the motility of the GI tract, for example, controlled by this system, and why you have some of the SLUD signs and symptoms. Okay, here we have a glimpse of a serine esterase, acetylcholinesterase in action. I told you about this molecular Hoover or vacuum cleaner, and we'll hope our technical system is working. And I can't see it on my screen, so I'll have to turn around and see if it's working. And the firefly-like objects that you see rushing into what's called the catalytic gorge on the way to the active site locus of this enzyme are representing acetylcholine molecules, but they could also be nerve gas molecules. They're attracted in the same way to the same active site. So they're rushing down this gorge inside this protein molecule toward the catalytic center of the molecule, where they will be torn apart and expelled as the component parts of this acetylcholine neurotransmitter. This enzyme is one of the most active, most efficient known in nature. It has what's called a very high turnover number. It can catalyze many thousands of acetylcholine molecules every second. I think it's maybe the second most efficient enzyme known. The steps of carrying out this reaction are in this bit of chemistry schematized here. Here I've reduced acetylcholinesterase, instead of this nice, elegant, beautiful protein molecule, I've just labeled it as ACHE, and there's this OH sticking out from the ACHE. This symbolizes the kind of business end of the molecule. In the catalytic center, there's a serine residue that has an activated hydroxyl group that is a very active nucleophile. That is, it can actually attack here acetylcholine at this carbonyl carbon and carry out this reaction of being acetylated. The enzyme is acetylated by acetylcholine, and then very rapidly it's hydrolyzed to release acetic acid and regenerate fresh enzyme. And this cycle takes place in about 80 microseconds, so very efficient, very fast. One thing I wanted to call to your attention is that there's first an interaction between the acetylcholine molecule and the active site of the enzyme that's reversible in nature. There's a K+1 and a reverse K-1 reaction. It's reversible at this stage, and this large dot symbolizes an electrostatic or maybe a van der Waals attraction between these molecules, but not a chemical bond forming yet. The chemical bond forms at a rate characterized by a rate constant called K2 that forms this acetylated molecule. And at this point, the acetylcholinesterase is actually inhibited. It can't operate on another molecule of acetylcholine until it gets rid of this acetate group on its active site serine hydroxyl. So this has to be hydrolyzed off, and luckily for us, this works, and it works very, very fast. In contrast, we have inhibition by the nerve gas serin. And by the way, nerve gas is a misnomer. It's not really a gas, it's a liquid, but like any liquid, it has a certain volatility, and it can go into the atmosphere and poison us as a gas. It can also poison us by direct contact as a liquid. So here's our acetylcholinesterase molecule again with the activated OH. The reason I show you all this chemistry is to show you the parallelism between the reactions of a nerve agent like serin and the natural neurotransmitter acetylcholine. They're exactly parallel. This was something that was hammered home by the work, classic work of Norman Aldridge at the Medical Research Council in the UK back in the 60s and 70s, where he showed that nerve agents and other organophosphorus compounds will interact with such enzymes as if they are substrates. They sort of trick the enzyme into behaving as though they're working on a substrate molecule. So here's the nerve agent serin, this organophosphorus compound with a fluoride leaving group. It has the same initial interaction as it had with the neurotransmitter, And here's this K2 reaction where it becomes acylated now at the active site serine, but now instead of acetate, it's an organophosphorus moiety on the active site of the enzyme. And this can be hydrolyzed off with a K3 reaction, just as we saw with acetylcholine in the normal situation. But in practice, with most organophosphorus compounds, and especially with sarin, this reaction, this K3 reaction, is extremely slow. So for all practical purposes, you're dead before the enzyme can be reactivated by hydrolysis. In the laboratory, you can measure this rate, but you're dead before the reaction can be carried out in vivo. We sometimes talk about the overall biomolecular rate constant of this reaction by looking at what happens to going from these reactants directly to this acylated product. And that's characterized by a rate constant called Ki, the biomolecular rate constant of inhibition. We have one other possibility that isn't possible with acetylcholine that we have with sarin, and that is this K4 reaction called the aging reaction. What happens here is, in the case of sarin, it's a dealkylation, or at least a net dealkylation, where we're left with this isopropyl group coming off the molecule, and we're left with a negatively charged phosphoryl moiety bonded to the active site of the acetylcholine. In this state, in the so-called aged state, it is truly irreversibly inhibited. We don't have agents that can reverse this reaction, and so once it's aged, you're really dead. If you weren't dead just from the inhibition, you're really dead when it's aged. Nerve agents are designed purposefully so that K4 is extremely fast, so that not only does the inhibition take place rapidly, the aging takes place rapidly, and the aging reaction obviates one possibility for therapy, which is using reactivation agents, some of the oximes like 2-PAM that can be used for ordinary insecticide poisoning and has some efficacy against nerve agent poisoning if it's administered extremely rapidly. But these reactions occur quite fast, and you generally don't have time to administer a reactivator. I want to get into some of the molecular modeling aspects of this that we're now getting into in our laboratory. I'm going to show you what the modified serine residue of acetylcholinesterase looks like in a molecular model that's actually been minimized for lowest energy state. Here we have the active site serine residue from acetylcholinesterase, and the rest of the acetylcholinesterase molecule has been blacked out, so we don't see what that looks like. We just see what I'm calling Waldo. And that's Waldo is a nickname for the active site serine residue from acetylcholinesterase that has this methylphosphonyl group, which is aged sarin. And so what does it look like in the molecule if we use the same sort of tinker toy model of acetylcholinesterase, which is a molecule of, instead of about 100, it's 65,000 molecular weight, very large molecule with thousands of atoms instead of just a few. And we have something like this. And so I ask you the question, where's Waldo? Does anyone see Waldo? If you have really good color vision, which I don't, my son spotted it immediately. I thought, wow. It's right here. Here's Waldo. And if we change the color on the rest of the molecule to sort of a steel gray, we can more easily make out Waldo down here in the active site, the catalytic center of the enzyme. And we can see it even better if we render, instead of tinker toys, if we render it in this beautiful manner, showing the secondary structures of the molecule, where we recognize certain regions that are alpha helices, and these arrows, these flat arrows, represent what are called beta strands. And here's Waldo lurking right here in the active site. So this tiny Waldo, this tiny methylphosphonyl group, can cause all this havoc. Just adding this one little chemical modification to this molecule inhibits it, blocks its function, and we have all of these signs and symptoms that I described from the Tokyo subway slide. So here, just to recap, we have acetylcholinesterase. Here is some organophosphorus compound, a phosphonate, where R could be anything, methyl groups, phenyl groups, And they will have different reactivities depending on what these groups are. But in general, here's what the reaction looks like. We have inhibited acetylcholinesterase. We now have inhibited an aged acetylcholinesterase. And I want to point out that when it's inhibited, we have cholinergic toxicity. And there are compounds like the carbamates that don't undergo aging, and they're just as toxic, and they can kill you just as dead if they don't undergo the aging reaction. So we have cholinergic neurotoxicity, but here it's treatable not only with the competitive antagonist atropine, which works at some of the synapses of acetylcholine, but not all of them, and oximes like 2-PAM that can reactivate. We can use both of these lines of therapy when the enzyme has not aged. And this will happen if you're poisoned with organophosphorus insecticides that don't age as fast as the nerve agents do. But if we have inhibited and aged acetylcholinesterase, we also have cholinergic toxicity, but now it's treatable only with atropine and with supportive therapy, such as being in an iron lung or a ventilator to ventilate for you because your intercostal muscles and your diaphragm muscles are paralyzed and you can't breathe. If we inhibit with something that can't age by virtue of its chemistry, it cannot undergo the aging reaction, such as this phosphonate molecule here. There's no aging, but we still have cholinergic neurotoxicity because that active site is blocked. The acetylcholine can't get to the active site, and this is still treatable with atropine and oximes. Now, I make a big point of this because it's going to be important for understanding the delayed neuropathic agents as we look at the parallel reactions that occur with the target enzyme that's involved in that case. And here we have the introduction to the neuropathic agents. This picture is of a bottle of the Jamaican ginger extract from the 1930s, late 1920s, early 1930s. Those of you who don't know about this, what happened was around that time, the US government, in all of its wisdom, decided to institute prohibition against alcoholic beverages, stamp out the evil of alcohol by simply banning it from all public consumption. You couldn't go to a bar and order a drink or have wine with dinner. You had to get your alcohol, if you needed it badly enough, from vegetable extracts, vanilla extract. One of the most popular during this time was ginger extract, and in particular the Jamaican ginger extract. If you can read the label, alcohol, 93%. It's pretty potent stuff. Well, the underworld got a hold of big shipments of Jamaica ginger, which came to be known as Jake, so, as a kind of a contraction of Jamaica ginger. And they started cutting it with anything they could find, shellac, varnish, various solvents. They were looking for things that didn't particularly have a bad taste, and you would have this nice gingery taste left by the Jamaica ginger. Well, one shipment, or maybe more than one, got cut with materials that had a compound called triorthocresyl phosphate, TOCP. They didn't know it until the epidemic happened, but this causes delayed neurotoxicity. Not a pharmacologic effect at the synapse, but an actual pathological effect at the axon, causing the axons to wither away and die, to undergo degeneration. And this is now known as organophosphorus compound-induced delayed neurotoxicity, or OPIDN for short. Some of the best literature on the effects of ginger Jake, of Jake leg, or Jake walk as it came to be called, comes to us not from the medical journals, because after all, people were drinking, they were breaking the law, and they were being immoral and breaking the law at the same time. And we have these blues songs that came up in the 1930s. And one of the best is from a group called the Allen Brothers. There's an old 78 recording that's been digitally remastered for your listening pleasure, which I can play right now, if it works. Sophisticated, it's a kazoo, guitar, and I don't know what else. Catchy though. Okay, well, you get the idea. I hear, you want to hear the rest? Okay. Maybe we'll hear the whole thing. I should have set this up as a karaoke.
Go for it.
No. Well, I'll tell you what. During the Q&A, if you want to hear the rest of the song, then we might do it then, because I'll need to get on with the rest of it. But let me just point out a couple of things. These lyrics are quite telling. I can't eat, I can't talk. Turns out there are lesions of the left recurrent laryngeal nerve. Now, normally, this disease affects the largest diameter fibers in the body, and it's symmetrical. But in the case of the laryngeal nerve, the left branch in humans is actually longer than the right branch, and you get this asymmetric effect. And it does affect your ability to swallow and to eat and to talk. But the main thing, the most apparent symptom and sign, is not being able to walk. There's a paralysis of spinal cord tracts and peripheral nerves to the legs, causing a paralysis, and there's also a sensory component to this. But the obvious thing is the motor component and the inability to walk. Down here in the spoken part, he says, "I'm feeling kind of drunk, brother." Be wearing Jake socks after a while. You know, they call them iron socks. And what they're talking about here, Jake socks and iron socks, were metal braces that people had to wear after getting this disease in order to be able to move around. Okay, we can talk about patterns of pathology that we see in the motor nervous system from different kinds of substances or diseases. There's something called a neuronopathy. First of all, on the left-hand side, just to orient you, is a cartoon of a normal nerve, normal neuron that has its cell body, its synapse, and it's going out to a muscle here, and these myelinating cells, Schwann cells, that produce the myelin, this sort of electrical tape insulator to prevent axonal crosstalk. If we have a neuronopathy, such as produced by, say, poliovirus or the disease ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, the neuron cell body dies, and so does its axon, and eventually so does its supporting myelin, and everything just dies, and we lose the connection between nerve cells and muscles, and there's a paralysis. In axonopathies induced by chemicals like the Jake leg chemical TOCP, we have a distal axonopathy. The distal end of the axon degenerates, and this may involve the lower third or maybe as much as half, and there will be some response in the cell body, and sometimes, in fact, the cell body will die as a result of this insult to the axon. And here comes back the idea of the dimensions, the basketball and the garden hose. And you can imagine that if you're this creature, this neuron, and you cut off half the axon, and that's maybe 90% of your body's volume, that's going to be a huge insult. The proteins and substances needed to replenish membranes that are worn out are all made up here in the cell body, and they have to be transported down this slender axon to where they're needed to repair things. So this may become just too much for the axon. There's now a school of thought that says that even diseases like ALS, which were thought to involve a primary attack on the cell body with a secondary axonopathy, might also involve an axonopathy even as a primary event, which would mean that studying triethylcressyl phosphate neuropathy might provide us with a good model system for studying ALS. In diseases like MS, you have an attack on the myelin, a focal attack on myelin, both in the peripheral and central nervous systems. And then in transmission toxicity, you're doing something to interrupt the action of the neurotransmitter, and that's what we saw with sarin. There's a glove and stocking distribution to this because of the largest diameter fibers being preferentially affected in these diseases. But even in things like the sensory neuropathy from diabetes, which is a metabolic neuropathy, we'll see this glove and stocking distribution. There'll be the use of this very sophisticated instrument in the neurologist's office. I'm not sure if I brought one with me, but a safety pin. You open up a safety pin and you stick it, does that hurt? Does that hurt? And so that's how they do these maps in a very sophisticated approach. And the motor impairment follows a similar pattern in OPIDN. Now, what does this look like in the laboratory? It was soon learned that the chicken was a very good model for studying ginger jake leg in the laboratory. And here is a control hen looking onto a hen that 15 days before the picture was taken was given a single dose of 2.8 milligrams per kilogram of a compound called di-N-butyl dichlorvos. It's related to the no pest strip pesticide, but it has a longer chain. Instead of dimethyl, it has a di-N-butyl, and this becomes neuropathic instead of being an anticholinesterase agent. Here's a control nerve from the tibial nerve, and here's a shot nerve from the hen in the treated state, showing these myelin ovoids where axons have essentially collapsed and the myelin is building up around, degenerated myelin is building up around the collapsed axon. There's an interesting structure-activity relationship of TOCP. This is TOCP, the triorthocresol. It has to undergo a hydroxylation reaction and a cyclization reaction to become the actual toxic material. It's actually the cyclic siliginin phosphate that's the toxic agent. And it became known early on that if you had tri-meta-cresol phosphate, if you put these in the meta position instead of the ortho position, it wasn't neuropathic at all. And if you put it in the para position, in the 4 position on each ring, it wasn't neuropathic at all. But in fact, if you have in this 3-ring system, if you have even one ring with a methyl group in the ortho position, you can form this kind of a product, and it will be neuropathic. It was also found that as we did more and more studies with different kinds of structures, that things broke down into type A and type B compounds, that we had compounds called phosphates, phosphonates, and phosphoramidates that if given in a sufficient dose to an unfortunate chicken, the chicken would become paralyzed. And there were other compounds, phosphinates, sulfonates, and carbamates, that were all known to interact with esterase-type targets, but these would never cause delayed neuropathy. However, if you pre-treated an animal with a certain dose of a phosphinate, a sulfonate, or a carbamate, it would protect the animal against a challenge dose of one of these compounds that would produce the delayed neuropathy. All of this was put together to discover the molecular target for delayed neuropathy. It's not acetylcholinesterase, but it's something that has certain similarities. It is an esterase, and it does interact through its nucleophilic OH group on organophosphorus compounds to become phosphorylated and aged. But the remarkable thing about this is that inhibition alone does not cause the disease. You can inhibit the enzyme with a phosphinate, for example. It can't undergo the aging reaction by virtue of its chemistry, and there's no OPIDN. And in fact, there's protection against OPIDN from a subsequent dose of a neuropathic compound. If you just get this far to the inhibited state, you do not get neuropathy. It absolutely requires the formation of this inhibited aged protein, and then you get OPIDN. Why? We don't know, because it's not as simple as blocking a metabolic pathway as it was in the case of acetylcholinesterase. It seems to be the formation of a modified protein. One theory is that this represents a signal, an aberrant pathological signal in the cell that signals the cell somehow to either give up maintaining its axon or actively to destroy it. Well, what is NTE? We don't know what it is, except we know something about its protein. We know the sequence of the protein now. It doesn't have a known physiological function at this point. The 3-dimensional structure is not known. But we do know that it produces OPIDN when chemically modified in a certain way by inhibition and aging. Recently, colleagues in the medical school in the neurology department, John Fink et al., have discovered mutations in the NTE molecule that are associated with ALS, or Lou Gehrig's disease. And here we have Lou Gehrig in his days with the New York Yankees before he developed this disease and then died shortly thereafter. Where our research is going now is we would like to be able to make the pure NTE molecule, the full-length NTE molecule. We've been able to make pieces of it to study. And working with Jeannie Stuckey and colleagues in the Life Science Institute in X-ray crystallography to get the three-dimensional structure. Quite often when we can get the three-dimensional structure of a protein, it will give us clues about how it works. We'd like to know its physiological function and its role in disease. What we have put together so far is work derived from computational molecular modeling, which has shown us that there is a transmembrane domain that goes through the endoplasmic reticulum membrane where this protein is found. There are cyclic nucleotide binding domains. There are 3 of them, and we don't know much about these yet. What we know most about is this region that we've modeled on the basis of a potato protein called patatin, and so it's called the pNTE, the patatin NTE domain. And we have a fragment of it that has been cloned out and produced in the laboratory called NEST, the NTE esterase domain, that includes the catalytic center. And so we've been doing a lot of studies on just the catalytic center, but I want to point out that there's much more to this molecule than just its catalytic center. One thing that's come out of these studies of the patatin domain, here's the potato, it's remarkable. Nature is always remarkable, but there should be significant sequence homology as you go through the protein databases of this mammalian nervous system protein with a protein that's found in abundance in potatoes. And so we've used this potato protein, patatin, as the template for instructing us how to build a three-dimensional model of NTE, or at least the active site domain region of NTE. One thing that's been discovered is that it has a catalytic dyad composed of a serine and an aspartate residue, as opposed to the more usual catalytic triad that's found classically in serine esterases and serine proteases. And there it is, looking inside this surface view of the molecule. Here, Sanjeeva Wijesakara in the lab has been doing virtual experiments with docking of a nerve gas-like compound, DFP. It's very much like sarin, but it has an additional isopropyl group. And he's docked this molecule into the computationally derived active site of our patatin NTE domain, and found that it nestles in this site very nicely with, and you can measure all of these bond lengths and angles and show that it's appropriate for what you would expect of an OP inhibitor in a catalytic region of an enzyme. We're also interested in the possibility that the chemical modification of this protein by these tiny nerve agents, these neuropathic agents in this case, could produce a conformational change in the structure of the molecule. And we wanted to see if computer predictions would bear this out. And we do see between the inhibited and the native state, if you look at the entire chain, this squiggle represents the alpha carbon backbone of the modeled protein. We do see a change that's propagated throughout. It's a small change, but it's propagated throughout the molecule. And we do see a rather larger change that is measured computationally in the active site region. Also, the same kind of thing has been done by simulating aging, by essentially taking the serine and sort of putting Waldo into the protein and then reminimizing the protein structure, and find that there is a difference between the native and the aged protein, three-dimensional structure. And again, this is predicted structure. It has to be confirmed experimentally using X-ray crystallography. We've now gone ahead, and there's another protein that's found instead of in potatoes, it's found in mammals. It's a cytosolic phospholipase A2 enzyme that also has sequence homology with the catalytic domain of NTE. And Sanjeeva has modeled this and found that, again, he comes up with the prediction of the catalytic dyad, and also another piece of the molecule that resembles something called a C2 domain in these phospholipases. So we're getting piece by piece more and more of this structure put together. And our ultimate goal is to get the entire structure and figure out what is the physiological role of this protein, how does it produce neurological disease, and is it related to horrific diseases such as ALS and other motor neuron diseases such as Stephen Hawking has. And I would just leave you with with these final words that echo some of Stephen Hawking's recent sentiments when he was asked, how can we survive the next 100 years on our planet with the advances of science and technology bringing us closer and closer to the brink with things like nuclear war that you'll hear about in a couple of weeks, or climate change, that we may already be past the so-called tipping point in that regard. But I'd like to leave this on a higher note, a note of hope, that we have chemical weapons, on the one hand, that can be put to all sorts of bad uses, obviously. That's what they're designed for. But they've also instructed us that science can be used, science and technology can be used for good, or it can be used for ill. Claude Bernard, in the middle of the 19th century, pioneered the use of toxins as tools to help the physiologist understand the inner workings of the cell, the basic physiology that makes our bodies tick. And he really pioneered this phrase that can be boiled down to using poisons as tools. So yes, they're horrific weapons, but we've learned much about the nervous system and how it works in its normal state from studying poisons, natural and man-made chemicals. We've learned a great deal about the nervous system by studying these actions. And I believe that, following in the steps of Claude Bernard, that seeking to understand these neuropathic agents and their targets like NTE will help us to understand neurological disease and ultimately to conquer it. Thank you.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program. Local broadcast is made possible in part by Pfizer.